In today's lecture, we will study Neo-Darwinism theory, and that is the last and fourth theory of evolution. In earlier lectures, we have studied the first three theories, that is Lamarckism, Darwinism theory, and mutation theory of D varies. And in this video, I'll discussing Neo-Darwinism theory. This theory is also known as theory of modern concept or modern theory, synthetic theory of evolution. This theory is believed to be the modified version of theory of natural selection, as the theory of natural selection was proposed by the Darwin, and this theory is given the name Neo-Darwinism from the name of Darwin, because this is the modified version of the theory of natural selection. This theory is reconciliation between Darwin's and de Vries theories. J.S. Huxley, R. A. Fisher, J.B.S. Halden, Wright, Ford, H. J. Muller, and T. Dobzhansky has contributed in the formation of this theory. Why this theory is important? What is the need of studying this modern theory? Because earlier three theories, they have given certain ideas for the evolution mechanism, but they were not 100% accepted. All the theories, they have certain points which are against those theories, but this theory of modern concept, that this is well accepted theory for the understanding of mechanism of evolution. Now, this theory is divided into three major postulates. This theory's first postulate is genetic variability, and the four causes are there for the understanding of the genetic variability. The second postulate is natural selection, and the last postulate is the reproductive isolation. Now we will study all these postulates one by one. Let us go through the first postulate, that is the genetic variability. As I have said that the genetic variability is the variability due to the changes in the genetics of the population. Now, this genetic variability can be due to mutation, genetic recombination, hybridization, or genetic drift. But anyhow, if there is a variation in the population that is due to the genetic causes, then it is known as the genetic variability. Now we will go through all these causes of genetic variability one by one. First of all, I'll discuss the first, that is the mutation. Now, as we know that mutation is of two types. First, chromosomal mutation, and second, gene mutation. Chromosome mutation is again of two types. If there are changes in the structures of the chromosome, then it is known as the structural chromosomal mutation, and if there is a change in the basic number of the chromosome, then it is regarded as the numerical chromosomal mutation. The structural mutation is again of four types: deletion, duplication, inversion, and translocation. What is deletion mutation? In deletion, a part of chromosome gets deleted from the main chromosome. The duplication is the duplication of certain part of the chromosome, that is also known as the addition, as that part gets added to the same chromosome again. What is inversion? Inversion is the process of cutting the particular portion of the chromosome that rotates at 180 degree, and again that portion gets rejoined to the same chromosome. In the translocation, a part of chromosome gets cut from the chromosome and it gets shifted on the another chromosome, that gets translocated to some other place on the other chromosome. The numerical chromosomal aberrations, there are of two types. One, aneuploidy. What is aneuploidy? The aneuploidy is restricted only to certain numbers of chromosomes, that is gain or loss of plus or minus one or two chromosomes. We can say that one chromosome may be added or it may be deleted, or two chromosomes may be added or deleted in the main genome. While the euploidy, that refers with the whole chromosome set. Let's say that we human beings, we have 23 pairs of chromosome, and in every pair we have two chromosomes. So in our genome we have 46 chromosomes. So if one set of chromosome gets added to it, then the numbers will become 69. So that process of addition of the one chromosome set or more chromosome set, or the deletion of the chromosome set, is known as the process of euploidy. Now we will study the gene mutation. The gene mutation is of three types. The gene mutation is also known as the point mutation, because here a part of a gene, that is the nucleotides, gets either added or deleted, or it may be replaced. So if a few nucleotides in a gene gets deleted, then the process is known as the deletion point mutation. If few nucleotides are added, then it is known as the addition mutation. And if the nucleotides are replaced, if the nitrogen bases are replaced, then it is known as the substitutional gene mutation. Now, the second cause of mutation is the genetic recombination. As we all know that the cell divisions is of two types. The first, that is the mitosis, and second, that is the meiosis. In mitosis there is no any gene recombination occur, but in meiosis there are chances of genetic recombinations. The two different kinds of genes gets recombine with each other, and we can have a newer combinations of the genes, and that genetic recombination occurs in the prophase 1 of the meiosis, and the main process during which the genetic recombination occurs is known as the crossing over. Due to the crossing over of the chromatids, we can have the new recombinations in the new generation, and that genetic recombination occurs in the germ cell, that are the microspore mother cell or the megaspore mother cell. The third cause of the mutation is the hybridization. What is hybridization? Hybridization is the formation of new species by combination of two different species which are genetically different. And the last cause of the mutation is the genetic drift. Now, what is genetic drift? The genetic drift is the elimination of some genes of original characteristic from the small population. Always the genetic drift occurs in the small population. When certain genes are lost forever, then that process is known as the genetic drift. This genetic drift can be due to epidemics. What are the epidemics? Certain widespread occurrence of the infectious disease. It may be due to the migration. Now we will go through the second postulate, and the second postulate of this theory is the natural selection. As we know that, what is the natural selection? The selection which is by the nature. But in Darwinism we have studied that the natural selection was on the basis of the fit organism. One which are fit can be selected by the nature, and one which are not fit, they were not selected, and that theory was considered as the survival of fittest, which was proposed by the Darwin. But in neo-Darwinism, that operates through the differential reproduction and comparative reproduction both. Now, what is the differential reproduction? The differential reproduction states that those members which are best adapted to the environment reproduce at a higher rate and produce more offspring than those which are less adapted. What does it mean? One which are fit, they can have the more efficient reproductive efficiency, and if they are having the more reproductive efficiency, then they are producing more progenies, and all those more progenies are also fit in nature, and subsequently for many generations, if they are producing the best adapted progenies to the environment, then we can have the newer group which is selected by the differential reproduction or the comparative reproduction. The third postulate of this theory is the reproductive isolation. Now, what is the reproductive isolation? The process of the reproductive isolation is very, very important, because in this we have to isolate certain species which are very similar to each other. So here, if the similar characteristics organisms, they are doing mating, and if they are reproducing the new progenies, then the chances of recombinations or chances of getting newer species gets decreased. Now if we want to increase the chances of new recombinations in the new progenies in the newer environment, then what do we have to do? The chances of the reproduction between the two different kind of organisms are must, and for that what do we have to do? We have to isolate the organisms which are genetically or phenotypically resembles with each others, and the process or the mechanism of the isolation of genetically alike organisms is known as the reproductive isolation. So the whole theory of neo-Darwinism gets divided into these three postulates. The first postulate was genetic variability, as we have discussed that these are the four causes of genetic variability. The second, natural selection, and the third is the reproductive isolation. So again we are revising the whole theory. The organisms which are genetically variable according to the differential environment, they can be considered as the best organisms to survive on this earth. So if they are best adapted to the for the survival, then those organisms are selected by the nature, and those organisms which is having the best adaptability, they have the higher reproduction rate, and due to the higher reproduction rate they are producing more progenies, and those characters get stable in the environment, and this is how we can have the new species which is fittest. And to reduce the chances of the similar characteristics organism production, what do we have to do? We have to isolate the organisms with similar characteristics, and that is known as the reproductive isolation. Thank you.